Force it because we keep giving him.
I'm not Michael. Okay, so
we should.
I just wanted to introduce Mike Hickerson. Mike Hickerson has just come in from CUNY, the City University of New York, in New York, where he's been for the last 8 years or so. So many of you already know Mike, so I'm going to make the introduction very brief because his record stands for itself. But Mike did his undergraduate at Evergreen College and then went on to do his PhD at Duke University with Cunningham. And then he came over here for a postdoc working with Craig Moritz. And we were here for 2 years or so.
Yeah.
And then took up his current position at CUNY. And so at CUNY, he's been tremendously successful. He's got a 5-year career grant at the moment that he's kind of in the middle of. And he's also got a Dimensions grant at the moment with Anna Carnival, who many of you also know, and so the Dimensions grant is focused on integrating different elements of biodiversity to understand dynamics in the coastal forest of Brazil. And so at the moment he's over here working with us to try and coordinate what's going on in the coastal forests of Brazil with what's going on in Hawaii. So Mike has been working, as many of you know, on a lot of different programs to look at how communities evolve over time using multiple different molecular markers. And some years ago he developed a program that many of you use, MS-Base. And Mike's going to talk about these approaches today. So thanks so much for coming, Mike.
All right, thank you, Rosie. Yeah, I didn't anticipate how much of a memory flashback this is going to be. I really have been here now about 8 or 9 years. Really happy to be here. Honored to see everybody again. So the title of my talk is Comparative Population Genomics for Historical Demographic Inference. And so what I'm really interested in is reconstructing history of biological and historical dynamics of biological communities, and that could be anything from whole regional assemblages down to sets of interacting species that are well characterized. And my approach generally is to develop models to make inferences about multi-species demographic histories. And the data that I normally focus on is population genetic data, what I try to refer to as population subgenomics, and I'll get to that later. And just sort of stepping back, one kind of inspiration here is Isaac Asimov, who had a trilogy called the Foundation Trilogy. And the idea here is that he had a character here who was interested in predicting the future. And of course, we're also somewhat interested in this in the context of global change. Of course, we're not going to really be able to do that, but what we can do is we can develop models, and we can test these models with real historical data, and to get a better idea about how historical forces have shaped biological communities. And so this is also inspiration here by Peter Turchin, who's sort of taken this approach to actually looking at real human histories. Interesting stuff. He refers to it as cliodynamics. Interesting stuff. But again, I just was really happy to come here back to the MVZ. I'm interested in reconstructing histories over space and time. And just to sort of step back here, I was here about 8 or 9 years ago with some vague ideas about developing Bayesian models for population genetic inference. And it was a really, really good environment, really good bunch of people here that really forced me to sort of formalize what I wanted to do. But just stepping back a little further here, kind of in a nostalgic mood here, but back in graduate school, there was a journal club that I'd go to, and there was a couple of papers looking at genetic divergences across the Isthmus of Panama. Now this is one of the most important sort of biogeographic systems, really, that we have on the planet. And so possibly maybe, further back in time than graduate school, but about 3 million years ago or older, depending on various estimates. The land bridge basically separated the community of marine species into basically twin communities. And this was, oh, here we go. You know, maybe this happened 3 million years ago, but see other, you know, more recent studies. And we could go a lot, you know, further back in time. But this is a really cool system for a lot of reasons. You know, we could ask questions about how sudden was this community isolation. Of course, there's the terrestrial end. What are the dynamics of invasion and community assembly from between North and South America? And also it provides an opportunity to estimate DNA mutation rates. And going back more than a century, of course, David Starr Jordan, this is a little local color here, he also noticed, you know, basically when you look at various regions, often sister species are found in neighboring geographical regions. For this very sort of reason here, often you'll have long-term sort of geological forces causing allopatric isolation. And this is a very clear-cut case. Now, when you actually look at genetic data, and this is going back, you know, 15 years ago in graduate school, there was a bunch of studies that came out looking at mitochondrial DNA between marine species, between Atlantic and Pacific marine species, and the general syndrome you would find was a pretty high level of variability between Atlantic and Pacific Ocean species. And at the time, you know, here's my graduate advisor, Cliff Cunningham. This is 1999, let's say. You know, what is he saying? Well, yeah, the mitochondrial divergences are really too variable for there to be a singular event. And he wasn't the only one saying this. Is like people in the lab and a couple of the other labs up, you know, back there in graduate school. But some of them still can be used directly to estimate mutation rates. Which ones? Would it be, you know, the more recent ones, the more older ones? And at the time, I took a class by Marcy Inuyama on coalescent theory, and I also was taking another class on ecological simulations, and the whole thing just didn't add up to me at the time. For one thing, and sort of one thing that maybe motivates science, I just had to bring in Joel Craycraft here in 1987. Anyway, what was pretty clear to me at the time is that coalescent theory would predict a high level of variability. We should just expect at the outset, even with the single divergence time across sister taxon pairs, we should expect quite a bit of variability at a single locus data like the like mitochondria. And you know, if you have different divergence times, you can often get the same amount of genetic divergence. But we should actually incorporate this amount of variability when making inference about mutation rates or divergence time or the dynamics of divergence across a whole community of species that may have colonized an area or have been diverged by a singular event. And again, I have an associateship with the American Museum, Natural History, so I see Joel all the time, and you know, we're always getting funny arguments and stuff like that. But this sort of older viewpoint is still quite pervasive, not just at the American Museum, but across the field in general. This is a study just a couple of years ago looking at a large number of taxa, terrestrial and marine, across the Isthmus of Panama, and both terrestrial and the marine divergence. So you have terrestrial invasion of South America from North America and vice versa, and you have allopatric divergence by the land bridge. And what these researchers found was that the connection between the two continents was actually a lot earlier, and this is consistent with recent geological evidence. Now, you know, take it or leave it of whether you believe that to be the case. This study didn't account for the coalescent. This is 15 years later, so it's kind of strange. Don't forget the ancestral polymorphisms. You know, this is something that's well known, well characterized, yet still there's sort of a kind of a disconnect. And the way I think about it is, some people see the rabbit, some people see the duck, and it's a little bit hard to see both at the same time. We've all been sort of brought up to thinking about the gene tree and to reconstruct the relationships across species according to the gene tree or the concatenated supermatrix without accounting for coalescent variability. It's a little bit, it's sort of a difficult, it's difficult to sort of take both ways of looking at the data at once. So going back to John Avis in the 1980s and the birth of the sort of phylogeographic revolution, this is a sort of a, even back then, this is like a well-known sort of aspect of genetic data. John Avis was well aware that genetic divergence was not the same thing as population divergence. But really, he was interested, you know, in a lot of different things and how, as far as how this data could be used. And one of the sort of larger questions that interested John Avis and his colleagues is that, well, we can easily collect this mitochondrial data and we can make inferences about whole communities. This was kind of a pipe dream back in the 1980s, and you know, you could really collect mitochondrial data with, you know, from anything, any animal species, really. And this has always interested me because it's really sort of bridging community ecology and population genetics. And so in this day and age, you know, we can actually get at these types of questions, I think, I'd like to think. So these days, these types of studies really come in sort of 3 different flavors, as far as I can see. Here's what I call, here's the whole genome approach. So this is using a sort of a very new method called PSMC, which accounts for divergence and recombinational information across whole genomes, and you really only need one genome per population to make very fine sort of resolved inferences about population history of particular populations. When you do this across multiple species, you can make inferences about how Earth's history may have affected the demographic histories of different species. This is a recent study, and you can see here's 5 taxa right here, and you can see that both of these species may have had a growth and a decline around at the same time. Another sort of flavor of approach for comparative population genetics is what I call the integrative approach. Here's a big study a few years ago, Lorenzen in 2011. So these types of studies, they use genetic data, not whole genome data, but usually just mitochondrial data, but also ancient DNA data, but also data from a lot of other sources, fossil pollen, paleo climate reconstructions, and so on. And then there's sort of a third approach, which I refer to as the mega mitochondrial approach. And this is like what I've been involved in quite a bit in the last few years. Here's a recent study looking at over 150 species pairs of birds that are more or less co-distributed across various barriers. So we have 3 riverine barriers. We have the northern Andes and Isthmus of Panama again. And in this particular study, we were interested in what was the sort of the dominant force of divergence in a very diverse group, in South American birds. Going back to these sort of classic hypotheses, is it divergence over emerging barriers? So is it like the Andes Mountains causing divergence? Or is it something more like dispersal over pre-existing barriers? And so we used a hierarchical Bayesian model that I developed here when I was a postdoc that basically treats all the demographic parameters across species as independent, but is able to estimate hyperparameters that describe sort of the summary history across your whole assemblage. And in this case, we're looking at sort of the variability and divergence times. Now going back to these 2 hypotheses, they have different predictions about the type of variability and divergence times. The general prediction is that under Under vicariance, we would expect dispersal ability. Well, in general, we would expect like there to be sort of several pulses of divergence, whereas under dispersal, we would expect more of a random stochastic pattern of divergences across barriers, more of like a sweepstakes colonization. In general, this is a lot of data here. We found much more support for the dispersal hypothesis, especially for all the riverine taxon pairs. However, there was a fair amount of signal for the vicariance hypothesis, especially for the Andean subset of species pairs. So a direction I've been moving this sort of what I call a mega mitochondrial approach is instead of models of divergence and colonization, I've been looking at models of expansion, and specifically the temporal dynamics of expansion. So, and sort of the general questions of how temporally synchronous or asynchronous are expansions into newly available habitat, and what is the sort of spatial patterns of expansion across whole sets of species. Do they come from the same area? Are there dual expansions from different numbers of areas within taxa? And so I just want to just kind of briefly go over an ongoing study I'm doing right now with Yvonne Chang and Frank Berberink, who's at is a new curator of herpetology at the American Museum of Natural History. For our data, we're looking at 90 lineages of vertebrates, all more or less co-distributed in the eastern United States forest region, and we're using different amounts of mitochondrial data. We're using again a hierarchical Bayesian model. Now one could actually make this type of inference individualistically across all the different data sets, and this is just sort of something that Frank whipped off right here. These are a large number of Bayesian skyline plots using the mitochondrial data, and as you can see, it's kind of a big mess here. And these types of studies, a little bit impractical for a large number of species, a little bit difficult to statistically interpret. The approach I like to take again is this hierarchical model. It enables formally testing various hypotheses using the entire data set, sort of all at once. And there's also arguably more statistical power because you're pooling across units that share parameters. So you're pooling the data across all your species that share parameters like expansion times, effective population sizes, divergence time, and so on and so forth. I'm not going to really dig into the nitty-gritty of the type of Bayesian models that we use, but just to take out two sort of important parts here. We have hyperparameters and species-specific parameters. Now these are allowed to vary independently across taxa, and these are basically summaries of the entire data set. In this case, we're looking at co-expansion, how synchronous was the co-expansion. So we have this hyperparameter here. This is the proportion of your assemblage that co-expanded at a single time. And then we have a co-expansion time. We also have independent expansion times and various other demographic parameters. And in this case, we have a very kind of, basically it's an expansion model, and you can think of synchronous expansion, everything starting from maybe a similar population size or a small population size, although they can be different small population sizes, and they can all expand at different magnitudes, but in this case, they all expanded at the same time. Here we have the sort of the opposite case, where they all expanded at different times, but you still have all these parameters that are independently varying across all your taxa. And then you can have the whole spectrum of intermediate cases where some things expanded in a single pulse, other things expanded idiosyncratically. You can have 2 pulses of expansion, 3 pulses of expansion. And I'll get to it later, but we've also expanded this to other demographies like contraction and so on and so forth, admixture. So what do the results say? This is an ongoing study right now. This is preliminary data, but it looks like there's a pretty strong case for congruence in especially the frogs. Here are the frogs right here. And this is the parameter that doesn't really quite come out here, but this is the proportion of species that co-expanded. So we have quite a bit of a strong magnitude for the frogs and the lizards, maybe the snakes too, not so much for the salamanders. And just to, you may be wondering, like, what do you actually call a species? And this is not, or a taxon. Really, this is a population-based model. So what we did is we, first of all, went through all the data, and as best we could, we subdivided things into panmictic units. So in some cases, we have a particular lizard species that's split into 3 different populations and then put in independently. Another thing you can get from this is these are posterior probabilities of the timing of co-expansion. And as you can see right here, snakes have a very strong pulse of co-expansion coinciding with the last glacial maximum. Mammals and frogs, really the glacial maximum before that, about 110,000 years ago. And very kind of diffuse inference for the salamanders and lizards. And now this is ongoing right now, and we're looking into what factors are really underlying these differences here. So moving on. So I've been mentioning these sort of mega mitochondrial models, and they're great because it's very cheap and easy, relatively easy to collect mitochondria from anything you want across large numbers of taxa. You have these big barcoding projects and so there's a lot of resources out there. But in the last couple of years, we've also gotten on the bandwagon of collecting so-called next-gen data, subgenomic data. And so we've been working on a couple of methods that make these types of inferences using things like GBS and RADseq. However, a lot of these methods are really flexible with regards to what type of data you have. You could have UCE data, exon data, or even just really messy, shotgun genome data. So the 2 projects I just want to highlight here. One is really focusing on time, the other one's space. This is a project by a graduate student in our group, Alexandra Jouet, and another project led by a postdoc, Diego Alvarado. So with the first project here, basically it's also a co-expansion model, but also we're incorporating co-contraction, and but it uses SNP data. So what's a good way to really summarize, analyze SNP data aggregated across species? Well, one thing we tried out, and it seems to work pretty well, is what we refer to as the aggregate site frequency spectrum. And just to really, and the type of data we're talking about, although this is, this is flexible, talking, let's say 5 to 100 populations, various numbers of individuals across populations, your run-of-the-mill SNP data set, 5,000, 10,000, 50,000 SNPs. And just to review, the site frequency spectrum, basically you're taking all your SNPs across your genome and you're categorizing them based on their frequency. So you have different frequency classes. You have SNPs where one variant is found only in one individual. You have another class where a particular allele variant is found in 2 individuals out of your entire sample, and all the way down to medium frequency and high frequency SNPs. So when you have multiple populations, one way to aggregate is to take all your singleton class SNPs, and just kind of, here we have them here in a sort of a toy data set of 5 species or 5 populations. In this particular species, we have pretty high number of singleton SNPs. Here we have a lower amount, or we have a very low amount. And what we do here is we reorder, we rank the singleton frequencies. And then we do this across every frequency class so that we, and one thing that really emerges from this is that we can sort of add all the data at once and have the summary of the Psych frequency spectra across all the data sets be independent of species or population identity. And with respect to the parameters we want to infer, this has a sort of a higher correlation with those parameters, which is good for inference. If this changes with respect to the parameters that we want to infer, then this is going to be useful for making estimates. So I mentioned we kind of expanded the model to account for contractions and expansions. We can have multiple pulses of both types of events across data sets, and we conducted, which is kind of a dangerous thing, a simulation study. If you, some people have simulation phobia, maybe that's a good thing, but in this case we didn't, and you can really go on forever. But we did learn a bunch of different things. But we basically we explored these different models here, these co-expansion, co-contraction models and mixed models. We used pseudo-observed data sets to see how well we could infer things using all these aggregate population genetic data sets. And we also compared different inferential frameworks. So I didn't mention what the actual inferential framework was for my hierarchical Bayesian model here was. In the past, I've been using approximate Bayesian computation, or more specifically, hierarchical approximate Bayesian computation. But we also, in this case, explored a couple of other frameworks, composite likelihood and random forests. We found, well, anyway, I'll get to that later. But just at the outset, we wanted to know whether the pooling effect actually helps. Do you get stronger inference the more taxa you add? And it's overwhelmingly yes. And even with moderate numbers of taxa, we get a fairly good discrimination power. Now this is just a visual summary. We're just using principal components analyses across the data sets, and just as sort of a handle of if things are working. We also did sort of formal cross-validation studies and under these three frameworks. We found that this one was best for our purposes. It was most tractable. It was quickest, and it led to the best, the most accurate estimates, also. And with composite likelihood, we're having some optimization problems also. Now, how about data? We first deployed this on a really well-characterized data set of three-spine stickleback. We wanted to use something where the history is pretty well known already. Now, this is the species, as you may know, colonized lakes in Alaska, only in the last glacial maximum, from source populations in the ocean. And so we used 5 population samples in this case, 2 from the ocean, and then three from different lakes in Alaska. And we just wanted to see how well it worked. This is a next-gen SNP data set, basically, and the results was a very high degree of co-expansion, as we expected, that coincided with the last glacial maxima. We searched temporal space going back to 100,000 years. And we did a lot of sort of cross-validation, again, simulation testing. We wanted to know whether this still worked when there was large differences in generation times, and it still works pretty well. When there's differences in number of SNPs across taxa, these are things that are really going to occur when you collect data, and it still worked pretty well. and also differences in number of individuals per taxon. Still seems to work pretty well. So moving on to another project, Diego Alvarado Serrano, he got his PhD in Lacey Knowles' lab at Michigan, and he's always been very interested in combining spatial and genomic genetic information. And so he's been with us for the last 2 years, and he's developed, one thing I'm not going to talk about, but he's developed a number of summary statistics. I don't know if there's any fans of Splash out there, but Splash is a kind of a pseudo spatially explicit coalescence simulator that allows you to make inference of demographic histories. But the information that goes into it is only genetic. There's no like explicit spatial information. So he thought that he could improve inference for Splash, but also just kind of in general by developing a number of summary statistics that account for geographical space. And that he found a number of them. But I'm going to focus on one right here that we refer to as the spatial Psych frequency spectrum. And here the goal is, in general, to locate the source of expansions, and in aggregate, when you aggregate across data sets, to locate the sources of co-expansion or what the spatial dynamics of expansion was in more complex scenarios. So it's analogous to the Psych frequency spectrum, but it captures information on relative allele frequencies and their explicit spatial distribution. And I'm just going to run through how it actually works. But just to give a little context, are there methods out there that allow for inference of the sort of spatial history populations? A lot of people just look at patterns of heterozygosity. So the prediction is that lower heterozygosity is going to be in areas that are founded by source areas. Then we have maybe patterns of FST. More recently, there's been this directionality index, which was developed by Ben Peter and Slatkin here at Berkeley. However, the spatial Psych frequency spectrum offers a couple of sort of advantages or maybe disadvantages, depending on your perspective and data sets you have on hand. But it doesn't require any outgroup. So this is SNP data, randomly, subgenomic data. You're randomly sampling or semi-randomly sampling, across the genome. But you don't have to make any assumption about polarity of your SNPs. You can have small sample sizes. So really, there is no population per se when you're actually constructing this sort of summary vector. You're really just taking the georeference information from an individual, a genotype. So it's really, the sample size is always 1. If you collected, I guess, 10 individuals under the same rock, they're all going to have the same georeference. But in essence, it doesn't have to be that way. And the way shared and private alleles uses all that information. So the way it works, this is a little funny, but you actually calculate the summary vector for every individual genotype in a data set. So we can just start from this individual here, individual 1, and you can think of these are different SNPs, and you have these two, the 2 alleles here for each SNP, and let's calculate the spatial Psych frequency spectrum for individual 1. So let's focus on that first SNP. Here we go. And we actually calculate the geographical distances between the alleles of that SNP and the other individuals. And in this case we have, and the color refers to the whether it's minor or major allele. In this case we have, let's just say it's both minor alleles. And so we actually we compile all the distances between that identical allele type. In this case it's only found in this individual 3, and we have 4 distances here. And then we just kind of, this maybe not be, there we go. Then we just kind of run across all the SNPs. Here's SNP number 2. We have a different bunch of distances here for this blue allele. It's only found in these 2 individuals, so we have these 3 distances, and you just kind of do the whole thing. And what you end up with here is, for each individual, you compile the spatial distances between copies of the minor allele across all loci that you have in your data set. So it's only the minor alleles, it's not the major alleles. And here we go. So you end up with something like this. And Diego's used Sturgis-based histogram to summarize this information. And what we were curious about is how this changes with respect to where the source populations are and where the the colonized populations are. So, when I mean populations, I mean that in not really the specific, explicit sense, because this could be a, sort of a spatially explicit model where you have discontinuity in population size and different densities and such. But the idea was that maybe there's some aspect of this that changes with respect to age of an area. And what we found is that irregularity actually is associated with distance from the source. And so I suggested that maybe we could sort of just summarize this, we could characterize this from Harping's raggedness index. This was originally developed for demographic inference of human populations, population expansion, so we're kind of retooling it for this purpose. And it seems to be a good way to characterize the shape of the distribution and its relationship with population age. And so Diego has been able to sort of spatially interpolate this information so that we can, once we have this calculated across all the individual genotypes, we can go back and basically estimate where the source ought to be, even given areas where we haven't sampled. And so Diego did another, again, large-scale simulation study to see how well it worked. First starting with the 1D model, 2D model, we tried single source histories, double source histories, so dual colonizations, and found that it worked fairly well. And so he actually used a simulator called FastSimCol2, which is a very flexible coalescence simulator, but it allows you to have a very large number of populations. So he's actually in our first pass here, he's simulating under a spatially implicit model where you have many different teams and you have different sort of migration rates based on distance and basically an isolation by distance model, but you have teams sort of originating in certain areas and then expanding out sequentially. And you have found there's a bunch of parameters here: expansion rate, migration rate, time since expansion. What did Diego find here? Well, so far we found there to be a very high accuracy and the ability to infer the source population. He's working on a way to infer 2 sources under a 2-source colonization model. We decided to deploy this on a data set of butterflies collected by Zach Gompert and colleagues. And these are butterflies in the western United States, so it's a really nice data set because they were sampled, diffusely across, different parts of the western states. We focused on 2 taxa here, one here close to home, and the others here in the Upper Rockies. And what did we find? So this is a species distribution model of, actually it's a Paleo species distribution model of these 2 taxa. So this is, as you know, we can make sort of retrojections of species ranges by correlating various environmental variables and species occurrences and what we think those environmental variables were in the past. And so we have this one that would look like it's kind of a habitat generalist. This is back in the LGM. It was found all over the place. This one here, kind of, it's predicted to have only occurred here in western Colorado. And so what did the spatial Psych frequency spectrum tell us? Well, in this case, it found that the source was over here. You know, who knows if that's, what that is really telling us, but in this case, it very sort of nicely told us that it was in the area of predicted suitability from the last glacial maximum. So, kind of just to wrap things up with a few things, you know. So I was here, you know, 10 years ago. This is like sort of the beginning when people really started playing with ecological niche models or species distribution models. And I had a beer with Bill Monahan, who was here, and we thought it'd be like a really good idea to try this on our friend Bigfoot. Bill kind of backed away from the project at some point, but Jeff Lozier was very enthusiastic about this whole thing, and so we decided to do it also on the black bear. This is a fun study. I just had to bring it up because for nostalgia purposes. There was a recent study on, what was it, unicorns or something?
Yeah.
Like, you know, global ecology, climate change, and looking at colonization of continental Europe from, anyway, yeah. So, a couple take-home messages. Back to the Diego's spatial Psych frequency spectrum. We're pretty hopeful about this, that it'll be a reliable tool for identifying range expansion, and ultimately, of course, we want to actually do this in aggregate across, you know, large numbers of data sets. You might be wondering, like, well, who the hell has, you know, 10, 20, 30, you know, RAD-seq data sets? Well, there are definitely some research groups that are collecting these type of data really at a large scale. I know that in Australia, Craig Moore's group, they're looking at collecting something like 40 to 50, you know, lizards, RAD-seq, and then there's another group in southern Australia. They're collecting this really on a large scale. And people, it's happening for sure. Again, it's ongoing work. The other study I highlighted. This other study I highlighted here. This is actually published. You can it's in the molecular ecology now. You can, you know, look up for the details. It's also Xander has. He's almost done with an R package that will be able to implement this stuff for mitochondrial and RAD-seq data, and it should be ready pretty soon. But so far it looks to be a pretty reliable tool for co-expansion. And of course, ultimately, I'd be interested in applying these types of models to sort of questions of community assembly and macroecology. And that's part of the reason why I'm visiting this week, is to sort of brainstorm ideas of sort of linking those types of models with these types of data, and sort of linking them by the hyperparameters that are in the models that we're playing around with. And just to really, again, I've been sort of, the last couple of years, kind of stuck in the Psych frequency spectrum. This is somebody who lived in Berkeley, I guess back in the 60s or so, Harry Smith. This is, I guess you could refer, this is kind of outreach, but about a year and a half ago, I thought it would be kind of cool to sort of retool the Psych frequency spectrum for looking at sort of quantifying dialect change between population samples. And it's actually not that hard to do. And we decided that our first data set would be this sort of a snapshot of the American vernacular, nearly 300 transcribed songs from the 1920s and 30s. And this is going to be, anyway, many of you are probably familiar with using phylogenetic tools. This has been done for a long time to understand language evolution. These methods in general use like basically the entire lexicon for your taxonomic units, whereas what we want to get at is something that can use the sample and that can be able to quantify demographic history or sort of language history at a more micro-evolutionary time scale. And so that's what we've been playing around with. This is just a Wordle thing, so you can kind of, it's already easily visualized here. And so, yeah, and what we've decided first is to compare two geographical regions, hillbilly, highland with lowland, and it's just a number of things stick out here. Here's a joint frequency spectrum of all the words, so we're using all the words found in all the songs. We didn't use the Cajun songs, or the instrumentals, of course. Well, you know, we have this one here and this one here, Lord and Jesus, but not sort of mutually frequent for some strange reason. And you can play around with this. This is going to be part of some liner notes in an upcoming CD release of. So Harry Smith, just going back to Harry Smith, he compiled the Anthology of American Folk Music in the 1950s. It's a very influential body of work, for the folk music revival. But he went through he collected 78s. He collected paper airplanes. He did a lot of different things. He was an experimental filmmaker, but he chose certain sides of the 78s. So what this release, what this new compilation is going to be, all the ones he didn't choose. So it's like the cutting room floor. But we're going to put this analysis in the liner notes. And then my friend Eli Smith, who's my partner in this, he found an old interview of Harry Smith from the early 60s, and apparently he tried to do this Psych frequency spectrum he transcribed by hand all the songs. Oh, whoops. Yeah. And he was, he did, but it was all lost. He was evicted from his apartment on the Upper East Side. But he wanted to know, whether certain words would kind of rise and fall with, before and after the stock market crash, when people refer to food after the crash and stuff like that. So we kind of, but this is after we already started the thing, so we were like, oh wow, that's a pretty wild coincidence, and let's try to look at those same things he was looking at. So, and that really, I really thank you for your time here. Happy to take any questions.
Psych frequency spectrum works with the singletons, and so are you throwing them out to this data set then, or is it need to be? It used to be that we would usually talk about a certain region having a high frequency of singletons, and you have an inference about that.
Yeah, I sort of glossed over some of the the tradition about how you read and how people use Psych frequency spectra. So it has different signatures consistent with various demographic histories. And the singleton class especially, it seems to be a good indication of history. However, with the methods of next gen RAD-seq and stuff like that, there's a lot of the singletons can be false positives and stuff. So we tried throwing them out, and it seems to work fairly well even after we threw them out. It didn't seem really matter much, but yeah, that's sensitive to that. Yeah.
For some of your SNP methods you were talking about, are they impervious to missing data, like particularly with RAD-seq, right, where you get this uneven coverage sometimes, or is it, have you looked into that? I mean, it can be real challenging.
There's sort of it's challenging to find that sweet spot, and so we've been having this debate at the museum about missing data. You have sort of one class of people who are just like, they don't want any missing data, complete data matrix. Yet they're missing a lot of data, because when you do that, you're not sampling a lot of SNPs, so you have low SNP coverage. And then I'm more like, don't worry about having the individuals. We want to sample the ancestors, but but you also want a good sample of the spectrum itself. So there's kind of a sweet spot there, and yet Xander has explored various levels of missing data, and it can allow for.
And with your spatial one, where you're calculating the distances, the matrix can take into account. You can have some empty cells.
That I'd have to refer you to Diego. I don't know if he's explored that yet, but the funny thing with the non-spatial one is that in a lot of cases we're forced to have missing data, because you'll have different numbers of individuals. So you kind of have to do sort of a rarefaction thing. You don't have to, but that's one way that works at least. You have to kind of go down to the lowest number of individuals you have. You know, you might have 50, 25, 10, but you tend to just sort of subsample 10 from everything, more or less. Oh, Jim, go ahead.
What's the source population for Sasquatch? You didn't tell us any results.
Gigantopithecus, my guy. Yeti. Yeah, we scooped Mark Siddall on that. Or was it, no, Karsten Raubach or something. Jeff is really tired of Sasquatch. He gets emails all the time.
Didn't realize what you were stepping into when you did that?
Yeah. You can have all those emails. Yeah?
Yeah, I had sort of a philosophical question about populations, and you know, a lot of us that work in musea are trying to figure out ways to use specimens so they don't get thrown away, and so whether or not you can call a group of accessions that were collected at various, you know, over periods of decades a population or not, and whether or not you could use those for RADTAG sequencing and so on, and infer anything from that at all, or I just want to know what your opinion is on that.
Are you referring to the sort of the puzzle of assigning genotypes to populations?
Yeah, or you know, whether you, I mean, obviously if you're sampling a group of specimens in a drawer, they were collected at various different times in history, but I know some people are treating them as, you know, because they're all from Nicaragua or they're all from whatever, their group of accession, they can treat them as populations. But they're not really true interbreeding populations. So there are people doing the possibility of RADTAG on these sort of data sets.
Well, yeah, I think it's great that we can do that, but I'm definitely of the school of, like, yeah, location doesn't equal population in the you know, the way the data works, and so you have to do a bunch of steps to help you assign genotypes to deans. And it's not always easy. It's never easy, or not always easy. Apparently, this R package, it basically it uses the FastSimCool2 coalescence simulator, and apparently that can use ancient DNA. You can have your tips be at different, you know, time slices. So that's good.
Yeah, so maybe you could actually time slice their specimens.
Yeah. I mean, there's always ghost populations. You know, that's another challenge, and you know, but probably a big factor in the way this data is generated.
So thank you, Mike. And if ever anyone wants to talk with Mike, Mike's going to be here for the next couple of days. So just yeah, you can just talk to me or talk to Mike. So thanks so much, Mike.